The overall goal of this procedure is to select and isolate chemoresistant ovarian cancer stem cells, or CSCs. This is accomplished by first fluorescently labeling ovarian cancer cell lines with red fluorescent protein, or RFP, and selecting for RFP-positive cells using fluorescence-activated cell sorting, or FACS. The second step of the procedure is to treat ovarian cancer cell lines with chemotherapeutic cisplatin for 72 hours, and then culture the surviving cells in CSC media. After 2 days, the non-adherent spheroids will form. Next, CSCs are characterized using gene expression analysis for stem cell markers, and CSCs are analyzed for cell surface markers using flow cytometry. Ultimately, analysis of CSCs for therapeutic response is achieved by treating cells with cisplatin or paclitaxel and performing an MTT assay to measure viability.
The main advantage of this technique over existing methods, like treating cancer cells with high doses of paclitaxel and cisplatin, is that our method yields more viable cells while using less toxic amounts of chemotherapeutic agents.
The implications of this technique extend towards improving therapy of ovarian cancer because CSCs represent a therapeutically resistant population that will need new treatment modalities. This method can also be applied to other systems such as pancreatic cancer, breast cancer, and other tumor types with therapeutically resistant cells.
Along with Jennifer, demonstrating this technique will be Dr. Lynn Roy, a postdoc from my laboratory.